Welcome to the fourth podcast from the Journal of the American Society of Nephrology. I'm Josie Briggs, editor of the journal. Joining me for this conversation is Mark Knepper, who's deputy editor of the journal, and at the NIH leads the Epithelial Systems Biology Laboratory at the Heart, Lung, Blood Institute. Mark, thanks a lot for making time for this, and thanks to your team for a terrific perspective essay that we're going to talk about today. The title of your article, Does SARS-CoV2 Infect the Kidney? This is actually a very hot debate in nephrology right now.
Yeah, thanks, Josie. It is a very hot topic. So with the advent of the COVID-19 pandemic, many of the basic science laboratories, including mine, have moved resources into projects designed to address the biology of the virus and host responses. I think more than a third of the intramural labs in my institute, NHLBI, have done so. And it's very exciting to see so many of our colleagues contributing their scientific expertise to the cause of stopping this terrible pandemic.
It is a terrible pandemic and getting worse right now every day. Let's start with the biology. Why has there been, since we first learned about this virus, a suspicion that the kidney might be a direct target of infection?
Well, there's very, very good evidence that the entry receptor for the SARS-CoV2 virus is a cell surface protein called ACE2, angiotensin converting enzyme 2. So we might expect then that cells in the body that express ACE2 would be targets for the virus. Unfortunately, this information was already available prior to the pandemic from multiple transcriptomic studies using next generation sequencing. And basically, those studies showed that the highest ACE2 levels are in the small intestine, number one, and kidney, number two. So what cells express ACE2 in the kidney? That's another question. A variety of labs, including those from Ben Humphreys and Cottle and Sustock, Andy McMahon, Mena Clatworthy, Anna Greca, and my own lab have contributed transcriptomic data showing predominant expression of ACE2 in proximal tubule cells. And immunocytochemistry seems to indicate that ACE2 is expressed mainly in the apical brush border of proximal tubule cells. So based on a lot of information, we think that the proximal tubule is a likely site of kidney infection by the SARS-CoV2 virus, and that the entry is likely to be from the tubule lumen.
Okay, so let's talk about what methods are available to actually demonstrate the presence of the virus in the kidney. Now, one of the first ways to look was electron microscopy, but that turned out not to be so simple, right?
Right. Not every 100 nanometer membrane-bound structure is a viral particle. So we have to be careful with the use of electron microscopy evidence alone to provide evidence for the infection. Fortunately, there are other ways to identify viral infection.
So what about RT-PCR or in situ to detect viral RNA? And what about detecting the viral protein by immuno? And will mass spec help?
Yeah. So basically, the SARS-CoV2 virus is an RNA virus, so it can be detected by reverse transcribing this RNA and amplifying it with PCR, that is RT-PCR. So RT-PCR is pretty good, and it's, of course, been highly optimized because it's the main tool we have to test for the disease in the population. A different way to detect viral RNA is through the use of in situ hybridization, which can directly demonstrate viral RNA inside of cells and is therefore more directly indicative of infection. An additional useful method is immunocytochemistry using antibodies to viral proteins. So antibodies recognize either the spike protein or the nucleocapsid protein of the virus are available commercially. However, immunocytochemistry can give false positive results, I think, as most of us have maybe experienced in our own labs, and really careful labeling controls are always essential in these studies. Perhaps the most convincing evidence for viral infection is recovery of the replicating virus from tissue via standard viral culture techniques. So there are a lot of tools that are available, and some are better than others.
Is it fair to say that recovery of live virus in urine or directly from kidney tissue will be the gold standard?
Yes, but it's always wise to use multiple methodologies, as done, for example, by the Huber group, to guard against false conclusions from systematic errors that are possible in any single method.
And given the methodological problems, where does this stand now?
So all of these methodologies have been employed in the various papers that were cited in our Perspectives article. It does seem that in some COVID-19 patients, the SARS virus has infected kidney epithelial cells. However, with the exception of one of the cited papers, observations have been made really in necropsy kidneys from patients who have died of COVID-19. So an important unanswered question, I think, is whether renal infection is unique to end-stage COVID-19, or whether it also occurs in less severe disease. If the latter is true, then we need to know how common kidney infection is as a function of disease severity. And this is important because acute kidney injury is a not infrequent concomitant of COVID-19, and it's possible that direct kidney infection is a contributing cause. So discovering whether renal infection precedes COVID-19 associated AKI would be very helpful in this regard.
Right. AKI is clearly a big problem in the hospitalized COVID patient group. So where do we need to go from here?
So we're only about 9 months into the pandemic, and many of the observations reported thus far are anecdotal, with observations in one or a small number of patients. And despite the advent of COVID-19 vaccines that are likely to come into play in the next few months, the disease is really likely to be around for quite some time. So I think we have time to do some of the type of studies that are necessary to truly understand the disease and its renal manifestations. So in my opinion, what we need is a scientifically rigorous approach with observations in large numbers of patients with statistical endpoints that we don't have yet. So this conclusion, I think, applies to this question that we addressed in the Perspectives article: when does SARS-CoV2 infect the kidney?
So keep going. How are we going to get definitive answers?
So I think that we can benefit from studies using ultrasensitive techniques to detect the virus in urine. If the SARS-CoV2 infection occurs in the proximal tubule of the kidney, or really for that matter in any renal tubule segment or podocytes, the basic biology of the disease predicts that the virus should be shed into the urine. Therefore, measurements of viral RNA, viral protein, or live virus in the urine should be capable of detecting viral infection in the kidney. Now, in our JASN narrative, we pointed out a couple of papers that looked for virus in urine, showing an extremely low frequency in hospitalized COVID-19 patients. However, these measurements were not really adequate to answer the question, in my opinion, because they really weren't optimized for sensitivity and could well represent false negatives. There are standard ways to concentrate viral particles, including ultracentrifugation. Furthermore, ribonucleases in urine can potentially degrade viral mRNA, leading again to false negative readouts. So on the positive side, meanwhile, some investigators have begun to exploit a new method, protein mass spectrometry, for the sensitive and specific detection of viral proteins. So I think it might be feasible to set up a fairly large study of hospitalized COVID-19 patients across the spectrum of disease severity using mass spectrometry-based viral protein detection and optimally viral culture techniques in ultracentrifugated samples to find out how common direct kidney infection is. And these studies should be done then with enough statistical power to be confident of the conclusions.
Okay, very helpful. To summarize and wrap up, we are convinced there's a need for well-powered studies with a representative population of hospitalized patients, maybe even some non-hospitalized patients, to determine the rate of viral shedding into the urine. Such data are critical if we're going to get to the base of understanding what is very clear, which is the high rates of acute kidney injury seen with hospitalized patients. By the way, we are planning a second podcast on that topic, AKI in COVID. Hope our listeners will tune in for that discussion. At this point, I think the data is not completely consistent. And as Mark has described, definitive methods have not been applied. Also, I would say right now it's fair to say that most reports seem to suggest that it's not simply infection, other factors that contribute to AKI, food volume and cytokine activation, inflammatory factors, particularly in the very ill patient. But clearly we know this is a very important problem. Another point Mark makes in his terrific perspective, which I urge you all to read, is that it will be important to know how frequently live virus is excreted in the urine in both hospitalized and non-hospitalized patients, because this is also needed to fully understand disease transmission. So important unsolved questions, but ones that I think we're getting closer to knowing how to approach. Thank you, Mark.
Yeah, I couldn't have said it better.
So thank you all, and tune in for our next podcast, which will again explore some of the important COVID-related questions in nephrology. Thank you all.
This podcast is copyrighted by the American Society of Nephrology, all rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare provider if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.